how much successful we are. So let's start with the taste. Another terminology for taste is gustation. And taste is detected by the taste buds in your tongue. Okay. So taste buds, if you see this is the dissection of your tongue, you have a taste pore, you have another taste pore here, and in the taste pore, you have extending taste hair cells of the receptors. So you see, you have a taste bud. In the bud, there are several taste cell or gustatory cells. These, uh, this one, gustatory cells, and some of the cells are supporting cells. Each taste cell is attached to the sensory nerve fibers. So when you put chemical compound here on the tongue surface, they either binds with the receptor or get inside this receptor. They depolarize and release neurotransmitter. That neurotransmitter binds with these nerve endings, and then they take impulse to your brain. Okay. So taste buds detect sweet, sour, salty, and bitter. And there is another special taste that is called amino acids or umami tests. Okay. So taste buds are modified epithelial cells with microvilli, which is the taste here. And each taste bud consists of 50 to 100 modified epithelial cells. Okay. So these cells, like in taste bud, there are several the epithelial cells, and each bud can respond to all categories of testosterone. That means in this pore, if you put sugar, salt, uh, the burned meat, or any bitter thing, they all will be tested here because all kinds of receptors are in each taste bud. Okay. So now, mechanism of taste. When you taste, what is the mechanism? How you taste it? So here are the 4 diagrams, and it explains how we taste salty, sour, sweet, and umami, and bitter. So let's think about it first. Salt is sodium chloride. So when you put it in your mouth, it becomes sodium and chloride ions. Shower has each chemical compound in the shower has hydrogen ion. Sweet is glucose, fructose, and galactose, so they are large molecules. Umami taste is for the amino acids, which are also large molecule. Bitter is quinine-like structure, large compound molecules. Okay, so each large molecules cannot get inside the receptor, but smaller ions like sodium and hydrogen can get inside the receptor cells through the channels. So let's see here. When you put salty on your tongue, then sodium through ion channel enter the hair cells receptor, and sodium is positive, so it depolarize. Once depolarize, then they open calcium channel. You remember from the release of neurotransmitter from last lecture. Once they depolarize, they fuse this vesicle containing neurotransmitter, which released here in the synapse that binds with the nerve terminal, and then sensory neuron takes the impulse away. Okay. So similarly, if it is sour substance, it contains hydrogen ion. Hydrogen ion enters and does the same thing, takes the impulse. But sweet, umami, and bitter, they are large molecule like sugar and quinine. They cannot enter through these channels. So they have to bind with the receptor found on the surface of these hairs. And then what they do, they activate another G protein, which lies under the plasma membrane. And then they dissociate the G protein. That G protein then activates second messenger like several kind of second calcium and other second messengers. And those secondary messenger then release calcium from the endoplasmic reticulum inside the receptor. And they, again, once the calcium comes increase in concentration, then what they do? They again fuse and release a neurotransmitter and text. So summary is salty and sour can cross and get inside the receptor, whereas sugar and quinine or umami, they bind with the receptor and activate second messenger, and you can taste it. So from the tongue, it goes to your brain in the temporal lobe where you taste it. Okay. Uh, Smell and olfaction is also the same way, like large molecule. So what happens? Smell is by your nose. On the surface roof of your nose, you see there is a olfactory epithelium in the cribriform palate of your ethmoid bone. And those receptors in the roof of your nose have several kind of receptors. You see, there is columnar epithelial cells, and in between them, there are olfactory neurons. You see, olfactory neurons and then epithelial cells. So these neurons have also here, cilia. So when you sniff it, the chemical compound comes here and binds with the receptor, and then they depolarize these bipolar cells and takes the impulse to your brain. Okay. So receptors are located in the olfactory epithelium at the top of your nose here. Uh, so in the olfactory, the apparatus, we call it olfactory apparatus. There are several kind of cells: receptor cells, supporting cells, and basal cells. So receptor cells receive the chemical compound. Supporting cells, when they die, these receptor die, these supporting cells, then again, sorry, the supporting cells then contains the detoxification enzymes, and the basal cells, when these hair cells receptor dies, they replace by mitosis of the basal cells. Okay, that's why when you have cold or some nasal infection, common cold, these cells like bipolar cells die, and then slowly the basal cell multiply and replace them again. This is the only place where, and some proof is that in the hippocampus, neurons can divide. Okay. So when you snip, they bind with the receptor and then takes the impulse. So how smell is, if I ask you between taste and smell, which one is more sensitive? It is smell, because smell has higher the uh, intensity. So even small amount of odorant you can smell, but a small amount of taste molecules you cannot taste. The reason is here. Other molecules bind to the receptors and act through G protein like quinine and sugar. But if you go back here, let's go back. So you see, let's see, this is your receptor in your nose. So when the odorant molecule binds with the receptor here, you see there is G protein. Each receptor is bound with several G protein in your nose. Let's go back. Up to 15 G protein may be associated with single receptor. In the tongue, there is only one association, 1-to-1, but here is 50 G protein. On top of that, once the odorant molecule binds with the receptor, there is dissociation of G protein, which release many G protein subunits. So you see, one is attached to 50, and then each further dissociate into several parts. So it is like magnifying. It's like fire. You light a fire and then fire ignite and become bigger. And that amplifies effect many times. that's why it leads to extreme sensitivity of a smell. That means you can smell a very small amount of odorant molecule. Human nose can detect, that's why, a billionth of ounce of perfume in air. Dog can even smell the smaller amount. That's why dogs are used to detect a lot of stuff. The reason is dogs' receptors in the nose are attached to more than 50 G protein, and their dissociation is further more magnified. That's why they smell even smaller, maybe trillionth ounce of perfume they can smell. Okay. Okay, so that is smell and taste in brief. Now let's talk about the hearing and balance. Okay. So ear has two functions. You can hear with your ear and you can balance your body. Which part of the ear balance and which part of the ear hears? We're going to talk about that. Okay. So before we go to anything, the hearing part or balance part, let's talk about the structure of the ear. So here you can see all the structure name here. So whole ear can be divided into three regions, three parts: outer ear, middle ear, and inner ear. So what are the boundary? The tympanic membrane or eardrum is the boundary. From here, everything outside is external ear. Helix, auricle, external auditory meatus, earlobe, these all are external. These are the external ear. From tympanic membrane to the oval window and round window or lateral wall of inner ear is called middle ear cavity. And middle ear cavity is also called tympanic cavity. Middle ear, it is middle ear which is connected to your throat through the auditory tube or Eustachian tube or pharyngotympanic tube, three names. And then everything from here inside your temporal bone, there is a twist and turn maze-like structure, cavity within a cavity, that is called inner ear. Another name for inner ear is labyrinth. So inner ear is inner ear equals labyrinth. That's why when you have infection of external ear, we call it otitis externa. Otitis means infection of the ear. When there is infection of the middle ear, then we call it otitis media. But when you have infection of the inner ear, we don't call it otitis interna. We call it labyrinthitis. Labyrinthitis. Okay. External ear and middle ear and inner ear all works for hearing. But only inner ear works for balance. Inner ear has three regions. You can see here, this coil region, the round region here, and then three half circle region. So this snail-like region is called cochlea. The middle round bump is called vestibule, and these three half canals are called semicircular canals. Okay, cochlea is for hearing, and these two are for balance. Got it? So this is the structure of the ear. We're going to focus first balance, and then we will talk about the hearing. So let's talk about the balance part, the vestibule and the cochlea. See? So from the vestibule and semicircular canal, the vestibular nerve goes to your brain, and from cochlea, cochlear nerve goes. And combined vestibule and cochlear nerve together is called vestibulocochlear nerve, which is eighth vestibulocochlear nerve. Okay. So let's focus on the vestibular apparatus. Vestibular apparatus is all hearing material in the inner ear. that is vestibule and semicircular canal. In the vestibule, you have two bump inside. You see, this is called saccule, which is nearby cochlea, and there is another bump that is utricle. So what these bumps contains? These bumps, saccule and utricle, contains hair cells receptors for balance. And then you have semicircular canal. In the semicircular canal, at the bottom of semicircular canal, there is a bump. Those bumps are called ampulla, and plural is ampullae. And these ampulla also contains receptor for semicircular canals. So when you move your body, the fluid inside these cavity moves the receptor and you feel your balance. Let me give you some background information. These structures, you have removed the skull part, the bony tube of the inner ear. So bony labyrinth is removed. This is the membranous labyrinth. Your ear is like a tube within a tube. Our textbook does not describe ear the very good way, because in your temporal bone, you have a maze cavity, and that cavity is like a twist and turn maze. And then inside that tube, you have a membrane tube, membranous tube. So if you have a tube within a tube, how you see tube within a tube? You have, let's see, two paper, and you make tube of two paper. So I'm going to make one a small tube, and I'm going to make another big tube. Let's see. And go. It's not sticking. Okay, so this is a big tube, and I'm putting another tube within a tube, small tube. You see? So outer tube, this tube is made up of bone, and this inner tube, can you see, guys? Can you see me?
Yes.
So
Yes.
outer tube is made up of bone, and this is called bony labyrinth, and we have removed bony labyrinth here. And inside this tube, there is another tube. This is the membranous, what you see in the diagram. So this is the membranous tube. So if I take this, you see there is a space between the outer tube and the inner tube, and then a space inside the inner tube. We're going to talk about that. Okay. So this membranous tube now, if I cut it, what do you see? You see there fluid inside it. So if you cut this cochlea, you're going to see fluid. If you cut this tube, you're going to see fluid. And that is called endolymph. Okay. Got that first? And you see all the auditory nerve is attached to the cochlea. All the vestibular nerves are attached to the vestibule and ampulla. Okay, so vestibular apparatus consists of otolith organs. Otolith organs are all these two structure. That is vestibule, which contains utricle and saccule, and semicircular canals, three semicircular, anterior, posterior, and lateral. These all structure provide sense of equilibrium, orientation to gravity, whether you are going up or down, or front or back. Okay. Vestibular apparatus and cochlea form inner ear. So you combine all of them, they are inner ear. Now I have here sensory structure located within membranous labyrinth. So you see, this is the membranous labyrinth, and inside the membranous labyrinth, we have sensory structure, which are the receptors. So this is filled with endolymph, and they are located within the bony labyrinth, which I explained earlier. So you have receptors here in saccule, utricle, and in the ampulla. And we are not talking about cochlea yet. So what is the function of these all balance? So what kind of balance is done by these things then? So Utricle and Saccule together is called again what? Vestibule. Okay. And the vestibule, which is Utricle and Saccule, provide info about linear acceleration. What is linear acceleration? It is like in line with your land. So linear acceleration is like when you are moving your body along the line of your horizontal, or you are going front and back, or you are going up and down. That is linear acceleration. Okay. Whereas semicircular canals oriented three planes give sense of angular acceleration. Angular means if you are moving your head, angulation, or you are moving, you are going, or your body is going angle wise, like somebody is doing a belly dance or moving your body, that is the angular motion. So you have to understand which structure has what kind of acceleration. Acceleration means movement. Okay. So how they do in the vestibule here in the receptor area, let's see here. This is the one of the receptor. So in the vestibular area, there is a receptor cell. On the receptor cell, plasma membrane is enfolding and they form stereocilia and one large kinocilium. So at rest, when you are standing or sitting quiet, what happens? At rest, they all are straight and it is surrounded by what? If you go back and check, they are surrounded by fluid. Inside it, you have endolymph. Now, when you move or you are in acceleration or deceleration or you are going up or down, what happens? They move. Okay. So when this hairs cilia moves towards the kinocilium, that means this is depressing the membrane and that is stimulating. And you see tiny lines, they are producing the action potential. So they are increased frequency of action potential and you can feel the position of your body. But it is away from the kinocilia back to normal, that is inhibited. And then again, slowly it backs to the rest. Okay. That's how the depolarization, hyperpolarization is going on and your brain can feel those depolarization, repolarization activity and you can feel. So hair cell are receptor for equilibrium. Each contains 20 to 50 hair-like extension called stereocilia. And one of these is a kinocilium. This is a true cilium. Okay. So we talked about this. And let's see an utricle and saccule then now. For the vertical or the uh, the linear acceleration. So this lady is standing here, head is upright. And when you have head upright, in utricle and saccule, you have the hair cells. You see hair cells? And hair cells are suspended in otolith organ. So otolith means calcium carbonate compound. They are like crystals of calcium carbonate. You see, otoliths are stone-like structure, rock-like structure. So they are stand still. Hair cells are stand still, and they all are attached to the nerve fibers. You see, sensory neurons. When she moves her head forward, what happens? This otolith, like mucus-like structure, they shag and then bend the stereocilia, stimulating the neuron, and that stimulates, and you depolarization goes to your brain, and you feel that your head is anteriorly. Okay. Uh, Sometimes some people get vertigo. Vertigo means you will feel moving your roof, you will feel nauseated, vomiting, and like your head will be spinning. Even you are not spinning your head. Do you know why that happens? Sometimes a small rock from here, they break off, and those rock then in fluid here start moving randomly. And when they move randomly, they hit hair cells. And when hair cell is hit, they bend. And that, like in the Fajard fashion, depolarize your hair cells and then takes the impulse to your brain. Even you are not moving, but depolarization is going to your brain, or depolarization and hyperpolarization, and that is causing like you're feeling all the time, and that is causing vertigo. Okay.
Professor, is that what also happens if you have many?
Yes. Yep. So a trickle is sensitive to horizontal acceleration, here pushed backward, you see, during the forward acceleration, and saccule is vertical acceleration. So this is vertical acceleration. Here is pushed upward when person descends. Semicircular canals, on the other hand, it contains crista ampullaris. They contain also hair cells, and then provide information about rotational or angular acceleration. They project in three different directions, like anterior, posterior. So how you move your head, and slightly anterior, middle, posterior, and based on that, like if you are moving your head, these all semicircular canals are depolarized, their hair cells, and you can feel the movement. For summary, you have to understand what is the function of each of these semicircular canal and the utricle and saccule, what kind of movements they're responsible for. Semicircular canal, you see angular. So when you move, what happens? Here is in semicircular canal, you have the hair cells, then you have endolymph of the semicircular canal, and there is cupula. Cupula is also like calcium carbonate, jelly-like fluid. So when you twist your head, move your head, what happens? It moves the fluid and jelly, and that moves the hair cells too. And sensory process bend in opposite direction of angular acceleration, and you can feel. Sometimes what happens, your eye balance with the movement, but sometimes our body does not follow the eye, and that called nystagmus. We do that test sometime, but this time we are not doing that test. So you can like move somebody in a speed and stop and then look at their eyes. They're like flicking one direction. That is nystagmus. Okay. So these all are basically when you move your head, you move the fluid, you move the hair cells, and that depolarize and that takes the impulse. Nystagmus and vertigo is the same thing I explained earlier. Vestibular nystagmus is involuntary acceleration of eyes that occur when spinning persons stop. So eyes continue to move in one direction opposite to spin, then jerk rapidly back to midline. So let's say you are moving somebody, there is fluid in the semicircular canal, and the vestibule is also moving. But when you stop somebody abruptly, what happens? Body stops, eye stops, but fluid is still moving. And that eyes try to balance that, and then eye flickers. That is called nystagmus. And vertigo, I told you earlier, is loss of equilibrium. This is the natural response to vestibular apparatus. Pathologically may be caused by anything that alters firing rate of eighth nerve, often caused by viral infection. So if sometime you have viral infection and affecting your eighth nerve, you can get it. Or as I told you earlier, that is called vertigo, the BPPV, benign paroxysmal positional vertigo. That is when you're a small piece of rock, like piece of the gelatinous material, break off and moves randomly in the fluid. Okay. So that is the part here. We talked about ears and outer ear. Let's talk about the structure of the cochlea now. Okay, so this is cochlea. We talked about vestibule and semicircular canal. Here we are cutting the cochlea in the middle, and then we see. So cochlea is, if you go here back, this cochlea is 1, 2, 3.5. So it is like 3.5 times coiled tube. Again, I said earlier, there is outer tube and there is inner tube. And if you coil this tube, how it looks like. So this is the tube, and then you coil the tube, how it looks. So this is the two tubes coiled together. And now you cutting here. So when you cut, what do you see? You see the outer tube, and then you see the inner tube. So in each, let me talk about it again. So if you have, why this is not working so nice. So if you put here the three tubes, and if I cut here, what I am going to see? I'm going to see one central cavity, another cavity, so two cavity. So if uncoiled this cochlea, then you will see three structure or three cavity structure. Let me make it simple and then I will come back here. So here you go. This is the cochlea, which is uncoiled. If I cut here or here, what I'm going to see? I'm going to see this space. This is called scala vestibuli. Then I'm going to see middle space, that is called scala media. And then this space is called scala tympani. And that exactly is there. See? So this is scala vestibuli, scala media, scala tympani. Okay. So one more time, how you hear when sound passes through this external acoustic meatus, it vibrates your tympanic membrane. And there are three pieces of the smallest bone in the body. They are called ossicles. From lateral to medial, MIS, malleus, incus, and stapes. Stapes is attached to the oval window, which is continuous with the cochlea. And then you have cochlea. Okay. So the tube is then divided into three fluid-filled chambers. Everywhere this will be three. So here is again tympani, media, vestibule. Tympani, media, and vestibuli, all of them in each coil. So only the media contains the endolymph. Scala vestibuli and scala tympani both contains perilymph. And we will talk about that again. Why? Here you go. So you see here, oval window is attached to the vestibuli. And then this tube goes all the way to the helicotrauma end of the cochlea, and then come back. And then it is continuous with another window that is called round window. So these two tubes, scala vestibuli and tympani, contains the perilymph, and media contains endolymph. And it is media where there is a basilar membrane, and you all lines here, those are the receptors, hairs. Okay, so let's go back and we will talk about oval window attached to the scala vestibuli at the base of cochlea. Vibration at the oval window induce pressure waves in perilymph, which is the fluid of scala vestibuli. Scala vestibuli and tympani are continuous at the apex. So waves in the vestibuli pass to tympanic and displace round window at base of cochlea, which is necessary because fluids are incompressible and waves would not be possible without round window. So if I go to again this uncoiled tube, so when sound wave comes, what happens? So when this download, when you go here and when the sound wave comes here, tympanic membrane moves, that moves the fluid, the bones, and that stapes then moves the fluid in the scala vestibula. You see arrow? And then it comes to the, because wave creates the energy, mechanical energy. Now this energy passes to the scala tympani, and that is released through the round window in the middle ear cavity again. But in the process of moving this fluid, it also moves the fluid inside the inner tube, which is called scala media. And in the scala media, there is hair cells, which moves the hair cells, and that takes the impulse. Okay. So, movement of basilar membrane induces. Basilar membrane is here. This is the membrane. This membrane is called basilar membrane. Here, see? All the hair cell lies over the basilar membrane. Okay. So movement of basilar membrane induced by vibration of the oval window produced a shearing force that causes the hairs to bend. Hairs means the receptors. And this opens potassium channel into the hair cell, allowing potassium to move down its electrochemical gradient from the endolymph into the hair cells and produce depolarization. Okay. This causes release of neurotransmitter that stimulate associated neurons of cranial nerve. So what happens when you hear sound in cochlea, moves the fluid, and inside the endolymph, the potassium ions are more compared to inside the cell. Usually what happens in other cells, potassium is more inside, but here in the endolymph, there is more potassium compared to inside the receptor cells. So potassium get inside. And that potassium depolarize that hair cell receptors. Okay. And then hair cell, due to depolarization, they release neurotransmitter that stimulates associated neuron of cranial nerve, which is eighth cranial nerve, cochlear nerve. Okay. And that's how you hear the sound. So now that impulse, we have two frequency of sound. So sound frequency is measured in hertz, you see. So if you have very shrill sound, that is like high frequency, 20,000 hertz. If you have a hoarse voice, like very rough, that is low frequency, like 50 hertz. So if you uncoil the cochlea, the high frequency sound stimulates the hair cell at the base of the cochlea, whereas low frequency sounds stimulates at the apex of the cochlea. Are you following me? Lower the pitch away from the base, higher the pitch near the base. Okay. Neural pathway for hearing. So how we hear when we hear. So let's start from the sound waves. Sound waves creates wave in the air, and then that air wave through your external ear is channelized to the external acoustic meatus and then to the tympanic membrane. From the tympanic membrane, the ossicles, from the ossicles to the oval window, then in the cochlea, from the cochlea, the cochlear vestibulocochlear nerve then takes the impulse, and then it goes to up to the medulla oblongata through the spinal cord. In the medulla oblongata, some of the fiber, you see, they cross, but some of the fiber goes to the same cell. And once cross the medulla oblongata, then it passes to the inferior colliculi. Inferior colliculi is the part of which part of the brain?
Quadra, quadrigemina.
That is the part of midbrain. Yes. And from there, like everything else, it goes through the medial geniculate nucleus of the thalamus. And from the thalamus, it goes to auditory cortex, which lies in your temporal lobe, where it hears the sound. And from there, it goes to auditory the Wernicke's area where it comprehend. That's how you hear the sound. Okay. Uh, Now the most common problem in the hearing is deafness. So deafness and hearing impairments. There are two kinds of deafness or hearing impairment. Be clear before you read anything. Pay attention. There is conduction deafness and sensory neural deafness. Sensory means neuron. So anything damage in the neuronal structure in the ear is sensory neural deafness. What can be damaged then? It is damage to the inner ear where there are hair cells, or cochlear nerve, or damage in the brain. So anything from inner ear to up to the brain is sensory neural or perceptive deafness. And any problem outside from there, like external acoustic meatus, wax, tympanic membrane rupture, uh, the fusion of the arthritis of the ossicles, those are the conduction deafness. Okay, so conduction deafness occurs when transmission of sound waves to oval window is impaired. Okay, and this impact all frequencies. Helped by hearing aids. If you put hearing aids on your mastoid process, then through the bony conduction, you can hear the sound. It has good treatment outcomes. Sensory neural or perceptive deafness is impaired transmission of nerve impulse and often impacts some pitches more than others. So if the hair cell is damaged towards the base, then high pitch sound will be gone. If you have to the apex, then low pitch sound will be gone. Are you following me? And it is helped by cochlear implants. Cochlear implants are put on the bone or inside, but it has not good result because you will work with whatever receptors are left by stimulation. You cannot stimulate all the cochlear nerve by the machine exactly like you are activating through your inner ear. So outcome is less. Okay. Now let's talk about the vision. Vision is the function of your eye. So eye. About the eye, I have gone over a little bit in your pre-lab learning PDF for your lab. And I have gone over pretty much a lot of stuff there. So if you have read that, that video, you can understand more here. Okay. So eye lies in your orbital cavity, which is formed by frontal bone, maxillary bone, and some facial bones. Okay. And only 1/6 of the eye you can see from outside. Most of the part is buried inside the cavity. Okay. If you take out eyes, this is the anterior pole of the eye, this is the posterior pole of the eye, and then this is the optic nerve coming out of the eye, which goes to your brain in the occipital lobe. So here we have a mid sagittal section of somebody's eye, and we are looking from the front side of the eye. Okay, from the lateral side of the eye, and you can see the wall of the eye. Okay, so this is front pole, this is posterior pole. A lot of information I'm going to talk here, so you need to pay attention about eye, pretty much all anatomy here. So eyes are attached to your orbital cavity, to the bones, through a lot of ligaments and very strong muscles. Those are called extrinsic eye muscles. They are superior rectus, inferior rectus, superior oblique, inferior oblique, lateral rectus, and medial rectus. Okay. Now, if you remove fat, muscles, everything, then you have an eyeball here. Eyeball has 3 layers. If you cut it, you can see 3 layers. The outermost layer is sclera, which is fibrous layer, and they are made up of fibrous connective tissue. The middle layer is choroid, and they are made up of the vascular connective tissue, a lot of blood vessels, a lot of melanocytes, and they are vascular layer. The third layer is yellow layer. You see deepest layer, that layer is called retina, and that is sensory neural layer, which contains photoreceptor to perceive or transduce the light energy. Okay. Now, this is the three layers all around here. But if you come to anterior pole of the eye, these three layers are modified and they have different names. So the names are the cornea, iris, pupil, lens. Those are the names. So let's follow. The sclera is opaque. That means light cannot pass through it. But when it comes anteriorly, follow me. When it comes anteriorly, it becomes transparent. And that transparent anteriorly region of the sclera is called cornea, and light enters through it. When you donate your eyes to somebody, you donate this cornea, nothing else. Okay. Now, choroid also comes here and then it modifies. It becomes, contains some ciliary muscle here. That ciliary muscle in the ciliary body attach zonular fibers of suspensory ligament, and that attached zonular fibers attach the lens to the ciliary body. Then some part of the choroid comes anteriorly and cover the lens, and this covering dark area of the eye is called iris. So this is iris. Iris is not complete, so there is central hole in the choroid here, and that defect hole is called pupil. Are you following me? The ciliary body and ciliary muscles inside it can contract or relax and change the diameter of the lens, which helps in accommodation. It can be thinner lens. Look at my hand. Thicker lens, thinner lens. Thick. So if the image is from the nearby, then it becomes thick, and you can see the, so that you can focus the image on the retina. We're going to talk about that. Okay. Now let's see inside the eyeball. Eyeball is not a solid structure. It has cavity, big cavity here, posterior to the lens, and this is called posterior segment. And then anterior to the lens is called anterior segment. Okay. Anterior segment contains watery fluid, and that is called aqueous humor. And posterior segment contains very thick jelly-like fluid, and that is called vitreous humor. Vitreous humor holds retina with the choroid on the wall. If somebody has less jelly here, in that case, retina will be detached from the choroid and there will be permanent blindness. Okay. So this segment is anterior segment and this segment is posterior segment. Anterior segment has two chambers. You see anterior chamber and posterior chamber. So anterior chamber is in front of iris and posterior chamber is behind the iris and up to the lens. So fluid made here and then made here and absorbed back here. Okay. Aqueous humor is produced and absorbed, but vitreous humor is permanent. Okay. Okay. So the around the iris there is another, the intrinsic eye muscles. They are a smooth eye muscles. So that is radial eye muscles and circular eye muscles. And these two muscles regulates the size of the pupil. So if there is bright light, dim light, then what happens? Your pupil dilates due to the contraction of radial muscles. And that works under the influence of the sympathetic nervous system. Where is bright light, then circular muscles under the parasympathetic nervous system constrict and reduce the size of pupil. So less light enter your eyes and still you can see the object. Okay. Now on the posterior wall of the eye, we have certain features, specific feature. This is optic nerve and this area is called optic disc. In this area, there is no photoreceptor. So we call this area blind spot. There is a pit here. The large pit area is called macula lutea and the small pit in the macula lutea is called fovea centralis. And fovea centralis contains only cones. So you see here, the large is macula lutea and the small dark is fovea centralis. So write down, macula lutea contains mostly cones and fovea centralis contains only cones. So if you want to see anything very clearly, the image must fall on this fovea centralis to see clearly. Okay. Refraction, your lens and cornea bend the light and focus it on your retina. That's why you focus image all the time on your retina. So, and that is the refraction property. So refraction is bending of light as it passes through media of different density, and refractive index is the speed of light in different medium. So let's see, refractive index of air is 1, which is compared to other media. Then cornea has 1.38 and lens has 1.4. So cornea will bend the light more than air but less than lens. How we determine what is the refractive index? That depends on the comparative density of two media. So when from air, like rays from air passing into your cornea, which is thicker, then it bends towards the center. When it is less, then it goes bifurcate more, diverge. And curvature of the interface between two media. So what is the curvature based on that? Lens provide fine control of adjusting focus because its curvature can be adjusted. Okay. So lens is the main organ inside your eye, which can change the curvature of the surface and focus the image on the retina by changing the bending capacity. You can see here, this is the image, falls on the cornea, you see bending, then again, it falls on the lens and then again bending and then get out of the lens, again bending and then falls on the fovea centralis. And what do you see? You see the image inverted and is small. And then your brain, again, re-justify it and you see exactly the size and shape of the structure. Field of vision. Each eye can see the object separately. Okay. Each eye can see the object separately and each eye has two visions, field of vision. The temporal and nasal. Temporal and nasal. So when the light falls on the cornea, the lateral or temporal side rays falls on the nasal side and nasal falls on the temporal side. And now the fiber from the nasal side, the optic nerve fibers cross and goes to other side of the brain. But fiber from the temporal side goes to the same side of the brain. That's why they are crossing. And this crossing area is optic chiasm. Your pituitary gland lies here. Okay. I think we need to stop here. Okay, so we have, I think 3-4 slides. You guys can work on it yourself. And I have talked about this in your another video that is in the lab video. I have talked about accommodation and everything there too. Okay. Thank you guys.
Thank you.
Thank you.
Professor.
you.
Thank you.
Thank you.
Thank you.